This video details the findings from what I learned investigating a study on why humans and other apes lost their tails. The article, Tail Loss in Humans, Apes Linked to Transposable Element Impacting Splicing, connects directly to my Week 4 and Week 5 investigations on transposable elements, alternative splicing, and genomic comparisons across species. The study looked at a key developmental gene called TBXT, which helps control tail formation in embryos. Researchers compared genomes from humans, apes, and old world monkeys and discovered something surprising. A single transposable element, specifically an ALU-Y element, inserted itself into one of the TBXT gene's introns about 25 million years ago. The small insertion had a major effect. Because there was another ALU element already nearby, the two elements paired in a way that changed the splicing pattern of TBXT. Instead of producing the usual protein needed to grow a tail, hominoids began producing a different splice variant that disrupted tail development altogether. Mouse experiments backed this up. When researchers edited mice to carry the ape-like version of TBXT, many developed short tails, or no tail at all. Some even developed neural tube defects, which suggests that the evolutionary pressure to lose the tail was strong enough to outweigh the risk. This study linked to our Week 4 investigation on transposable elements. We learnt that ALU elements are a type of sign retrotransposon, a piece of DNA capable of copying itself and inserting itself into new positions in the genome. Even though most transposons in humans are now inactive, ancient insertions remain highly influential on our current phenotypes. What stood out is how a non-coding transposable element insertion can change gene expression without altering protein coding sequences. This matches lecture content showing that transposons often affect genomes not by adding new genes, but by creating structural changes, altering regulatory regions, and changing splicing patterns. The study used comparative genomics and tools like BLAST to identify conserved and divergent sequences across species. This study relied heavily on comparing orthologous genes between primates to pinpoint a change unique to apes. Because all hominoids share the ALU insertion, but old world monkeys do not, the study could infer both the timing and the evolutionary significance of the mutation using these tools. The study's strongest feature is its use of multiple approaches: comparative genomics, evolutionary timing, and CRISPR mouse models, which together provide robust evidence linking the ALU insertion to tail loss. An interconnected approach improves the validity of the study. A key limitation is that the study cannot determine whether the ALU insertion was the sole cause of tail loss. Other genetic or regulatory changes may also have contributed, and the mouse experiments showed incomplete consistency. This investigation led me to want to explore more about how non-coding DNA shapes human evolution, especially through mechanisms like splicing and chromatin regulation. I am also now interested in how researchers use genome comparisons to reconstruct evolutionary histories. Overall, this study helped me see how molecular changes in transposable elements scale to evolutionary outcomes and how the junk DNA we often overlook can drive some of the most important traits in our lineage.